So next we have Dr. Change Tan. And let me find her, and we'll be on our way. Here she is. Okay. 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 Hey, Change.
Yes.
Good morning.
Good morning. Hi.
Wonderful to have you with us. Let me just briefly introduce you, and we will give you the floor. Let's see. Okay, so Dr. Tan received a BS in chemistry from Hunan Normal University, an MS in organic chemistry from Nankai University, and a PhD in biochemistry from the University of Pennsylvania, and also completed postdoctoral training in genetics at Harvard Medical School. She is currently an associate professor of biological sciences at the University of Missouri. She co-authored the recent book, The Stairway to Life, An Origin of Life Reality Check with Rob Stadler, who I believe is with us today. Dr. Tan's talk today is entitled, If Darwin Were Alive Today. Change, please come and share with us.
Good morning, everybody. I'm so happy to, hear what Rob just said about the 4 dimensions of a human genome. I wonder if you have ever wondered where did we come from and how we are related to the bugs in our gut, the food we eat, the flowers we love, and the animals we adore or am afraid. Is it that men, as a Hungarian proverb says, men are carried by horses, fed by cattle, clothed by sheep, defended by dogs, imitated by monkeys, and eaten by worms? Or shall we change it to viruses? Or is that man and all other life forms are connected, as Darwin said, via one or maybe a few evolutionary tree of life? Well, actually, how many trees were in Darwin's mind? Is that one or several? Well, he, in his conclusion page, he did not make that clear. Is that maybe, originally breathed into a few forms or into one? But earlier he said he believed that animals have descended from at most only 4 or 5 progenitors, and plants from an equal or lesser number. And he continued that with analogy, he said maybe all animals and plants have descended from some one prototype. So the conclusion was that all the organic beings which have ever lived on this earth have descended from some one primordial form into which life was first breathed. And from that one form, one life form, species are produced and exterminated by slowing, acting, and still existing causes. Not by miraculous acts of creation and by catastrophes, but by mutual relationship of organisms to organism. The improvement of one being entailing the improvement or the extermination of others. Where did Darwin really believe that the first life was breathed into being? From his letter to his friend Hooker, seems the answer was no. You know, he imagined that in some warm little pond with all sorts of ammonia and phosphoratic salts, and then a protein, you know, compound was chemically formed, ready to undergo still more complex changes. That is evolution. So I think it is fair to say that in Darwin's mind, really life came from no life, and a complicated life evolved from simple life, while mutation or variation and natural selection and a long time, where this is what I believed for most of my life too. Darwin, he also addressed the question, you know, why have all the most eminent living naturalists in his day and geologists rejected his view of mutability of species? Where he said, because actually there are legitimate objections and difficulties that can be urged against his theory. But he said that the more important objections related to questions on which they were ignorant. And he also said that the chief reason, you know, people are not willing to accept his idea was that we are, quote, we are always slow in admitting any great change of which we do not see the intermediate steps. Nobody have ever seen those intermediate steps. He also said that, you know, people tend to hide their ignorance. So really, how ignorant were Darwin and his contemporaries about life and the relationships of different life forms molecularly? Where Darwin published his Origin of Species in 1958, and we are 2020, right? Almost the end of the 2020. 10 years after the publication of Origin of Species, DNA was isolated. However, DNA was not regarded as the genetic material until more than 90 years after the publication of Origin of Species. And at about the same time, with Watson and Crick figured out the double helical structure of DNA. So Darwin and his contemporaries were ignorant about the genes, what genes are made of, not say, you know, 4-dimensional, they even have no idea about the 1 dimension. Well, how have I got involved in the issues of the origins of life and of biodiversity? You know, as I mentioned earlier, for most of my life, I believe that life came from no life and complicated life from simple life, while mutation, natural selection, and time. Well, in 2006, George Smith, he asked me to teach molecular biology, a course I did not plan to teach because I had never taken the course myself and I had to self-teach. Well, that request from George Smith changed my life entirely, unknowing to him and unknowing to me. Well, I learned from molecular textbooks and journal articles that actually eukaryotic cells can't use the DNA replication, transcription, translation machinery of bacteria to replicate or transcribe or translate their genomic DNAs and vice versa. For example, here I have DNA replication initiation in E. coli on the left, and those for DNA replication initiation in yeast. You can see actually all those proteins involved. There are many. Actually, E. coli, has dozens of genes involved in DNA replication. And yeast has over 100 different proteins involved. These proteins involved are mostly unrelated. They are unrelated in sequence at all. So I wonder if we can just call that the paradox of organism-specific genetic information coding and decoding. Another thing I learned was that most essential genes are taxonomically restricted. So here on the left, I have 4 different bacteria, and on the right, 5 different eukaryotes. We're looking at distribution of essential genes. And you'll see that the vast majority of bacterial essential genes are bacterial domain specific. They do not have genes similar in eukaryote. And the vast majority of eukaryote essential genes are eukaryote domain specific. So you do not find any homologs in bacteria. May we call this the paradox of organism-specific essential genes. So this really poses an insurmountable challenge for the evolutionary origin of eukaryotes. The idea that eukaryotes had evolved from prokaryotes. In 2010, Craig Venter published an article, and he gave a talk. He described the first self-replicating species we had on the planet, whose parent is a computer. And in that synthetic bacteria, he actually embedded a quote, say, "What I cannot build, I cannot understand." Well, inspired by his work, I decided to approach molecular biology, teaching molecular biology, from the perspective of synthetic life, and I call it understanding life by building it. So synthetic life really is at the crossroad of chemistry and biology. Where to understand life while building it, we needed to ask what to build, how to build it, what problems we encounter, and what solution do we have, right? Solutions by cells and solutions by humans. So we can, just divide the stairway to life into, I have 6 steps here instead of the 12 step in the book we described, Rob and I wrote. So the first step is the formation of basic building blocks of life. The next step is linking those basic building blocks together to make biopolymers. And the next step is making biopolymers that function like a cellular DNA, RNA, or proteins, in short, genes. The next step is making genes with a required function. And then the following step is making, genes into a living cell. The final step is making a complicated cell from a simple one. We have just discussed or talked about that, actually the idea of complicated cells like eukaryote evolved from simpler ones, the prokaryotes, including bacteria and archaea, are problematic because of organism-specific genetic information coding and decoding, and organism-specific essential genes. So I'm not going to talk about that anymore, right? About formation of basic building blocks of life and formation of biopolymers. Well, imagine if the vendor team had to start from there, starting from making basic building blocks, and then using those building blocks to make biopolymers. They would encounter a few paradoxes, right? The paradoxes of asphalt, homochiral, and water, and homolinkage. Well, these paradoxes, these problems actually are humanly solvable, solving not by really solving it, but by bypassing them. So vendors team did not, start from that. And so I'm not going to talk about that either, right? If you're interested, you can go to read the book Rob and I wrote, talking about 12 steps from chemistry to biology. Of course, none of those steps were visible or knowable to Darwin. So that's part of the ignorant he and his contemporaries had. Right, so vendors team actually, have to start from the formation of biopolymers that function like cellular DNA, RNA, proteins, or genes. Well, actually, they did not even start from there. But if they did, they would be pushing the wall. Because here they would encounter the problem of information paradox of life. And this is not solvable humanly. There is no bypass. The information paradox of life is the absolute necessity of particular sequences of nucleotides in DNAs and RNAs and amino acids in proteins for their functionality, and the absolute lack of any chemical or physical laws apart from living organisms to account for the sequences. So on the one hand, for a gene to have a specific function, the nucleotides it contains have to be arranged in a special sequence. On the other hand, for the gene to have a specific function, the sequences of the nucleotides must be free to change as required by its function, not by the chemical or physical affinity of the nucleotides. So we need to be free and not free at the same time. This applies for DNA, RNA, and proteins. This is very similar to our language. For a sentence to have a specific meaning, the letters it contains have to be arranged in a special sequence. And for a sentence to have a specific meaning, the sequence of letters of that sentence should be free to change as required by its meaning, not by the chemical or physical affinities of the letters. Right? If I say Tom had a fish for breakfast, that's good. But if a fish had Tom for breakfast, that's a totally different story, right? The same letters arranged in just a different way, you know, we have different meanings. Actually, for the vast majority of the arrangement of the same letters will be gibberish. So like the sequence of DNA, RNA, and proteins and their function, language, you know, letters of the language and the words and the sentences of the languages, right? We need to be free and not free at the same time. So this is the information paradox of life. Well, what is the solution, right? For Vander's team, their goal is to build a self-replicating cell. So what solution do they have? There's no human bypasses. The only thing they can do is to borrow from existing cells. But what to borrow? Right, not just any gene. You need genes to have required functions. We know essential genes are important, right? Because an essential gene is necessary for the viability of the organism. The organism contains it. So we need essential genes. Well, this is actually demonstrated by Vander's cloning experiment, right? It happens that in their cloning process, they had the misfortune of, having one single base pair deletion in one essential gene, DNA gene. So that actually DNA gene is the protein involved in the first step in the recognition of orange replication in bacteria, right? One single base pair deletion in a meaning base pair genome rendered their cloning experiment not working. they needed to add back that deleted one base pair. So we need essential genes, and there are no bypass. Well, actually, studies of essential genes find that even for the simplest bacterial cell, hundreds of genes are required for the survival and propagation. So basically, even the simplest cells, there are hundreds of essential genes are required, right? So not only that we need one essential gene, actually we need many. We need many essential genes that perform specific functions, and there's no bypass. Some of the essential genes, we know their function. For example, genes involved in DNA replication, transcription, and translation, and genes involved in antigen harnessing, biopolymer repairing, selective transport of nutrient and waste, and means for gene regulation. But we also have essential genes with unknown function. That's troublesome, right? So actually, after Vander's team made their first self-replicating bacterial cell, they went on to get rid of as many genes as they could to figure out, what is a minimal requirement for a self-replicating cell. And they find that, so that is their 3A version, right? The first one is a 1.0 version. They find that there are still, in their 3A version, there are still many genes of unknown function. Well, for essential genes with no function, well, we know what to borrow. With essential genes with unknown function, we don't even know what to borrow anymore. So the solution is to copy the whole thing, right? To borrow the whole genome of an existing cell. That's molecular cloning. By the way, the goal of Vander's cloning experiment or making the self-replicating bacterial cell is not to study the origin of life or origin of biodiversity. But their experiment really is very reviewing for the origin of life and the origin of biodiversity. It forced people to think about the origin of life and the origin of biodiversity from the imaginary to the reality. So to make their self-replicating bacterial cell, actually it's mycoplasma mycoids. They started with an existing mycoplasma mycoids, and they inserted in this genome a yeast cloning vector to facilitate their later cloning procedures. And then they isolate the genome of the bacterial, sequence that, and inserted 4 watermarks, including one with a quote, what I cannot build, I cannot understand, right? So then they synthesize the genome, And actually the whole genome was assembled in yeast. And they can make many copies of the genome in yeast. And they can isolate the genome, many copies of them from the yeast genome, but they cannot make a self-replicating bacterial cell from the yeast cell, a eukaryote. They cannot make a self-replicating bacterial cell from the genomic DNA that isolated from the yeast either. Well, the bacterial genome was cloned and assembled in yeast cells, but the yeast cells cannot make any bacterial cell using the genome, even though yeast cell contains many, many more proteins and many, many more RNAs than the bacterial cell. Why the yeast cell cannot give birth a bacterial cell? That's because DNA, however long it is, however much information it can code, is useless unless its coded information can be interpreted and executed by its matching or cognate proteins and RNAs. Eukaryote cells can't actually, transcribe and translate the bacterial genes, right? You have a different decoding system. So to make the self-replicating bacterial cell, the vendor team had to transfer the bacterial genome into a host cell, a bacterial host cell, a bacterial that is very, very similar to the genome donor. And then they needed to separate the cells that contain the synthesized genome from the cells with its own genome, the endogenous genome. So we see that, not only we have insurmountable challenge of making eukaryotes from bacteria via natural means, we also actually have insurmountable challenge of making bacteria from eukaryotes via natural means. So you cannot go from simple to complicated, and you cannot simplify the complicated, streamlined, and make the simplified, right? I hope that I have also demonstrated to you there is an insurmountable challenge for Orange of Life via natural means. Because of organism-specific genetic information coding and decoding, not only you need a coding system, and you also need a decoding system at the same time. Even you have a 4D genome, you need something to decode the genome. And that decoding system and the coding system have to match with each other. Furthermore, in addition to the coding and the decoding system, which are organism specific, we have other genes, other essential genes are organism specific, right? So really the idea of a one big evolutionary tree of life does not hold water. Well, many people have realized that and are promoting the idea of a web of life. But I think the idea of a web of life does not hold water either, because the idea of a web of life or a web of life can be an artifact of forcing a forest of tree into one evolutionary tree of life. Not only we have a forest of life, I think life actually is modular, and different life forms can share one or multiple modules. So we see that actually when we look at organisms around of us, we see a lot of similarities, right? Actually, Darwin, developed his theory of common descent by natural selection, by looking at those similarities. But we also have discontinuities among different life forms, which more and more data are proving. And we also start to see the modularity of life. Actually, I'm going to discuss this afternoon about the three pillars of biospecies, right? Similarity, discontinuity, and modularity. Well, if Darwin knew what we know today, or if Darwin were alive today, I think he will have a major revision of his origin of species, and to come to the same conclusion as I have, which instead of one big evolutionary tree of life, we have a forest of life, and indeed, what we have, the evidence of genes and genomes are consistent with what the Bible teaches. God created plants and animals according to their kinds. I also think that Darwin would, revise his book of Descent of Man, which really, clearly say Black life matters not. In his mind, Black lives and, yellow life too, Chinese, right? Oriental nations, right? Were savages in his mind. So he would need to, have a major revision of his Descent of Man. He would come to the same conclusion because of human-specific genes, right? Genes that have no homologs in any other life forms. He would come to the same conclusion as I have, that it's impossible for human to be evolved from animals, as Darwin's Descent of Man's book, says, right? So he might come to the conclusion that really here too, right? As the Bible says, Black life matter, do matter. Black life does matters, right? Because actually all men were created in the image of God. God is correct, as Dan said earlier, right? He is right about history, he is right about biology, about the relationship of different organisms and humans. Thank you for your patience. What should I do? Okay.
Thank you, Tan. If just stop share. There you go. Thank you.
Okay.
That was awesome. Let's see if anyone has a question. Okay, I'm going to ask a question then. So I remember from reading your book, you're talking about the RNA world hypothesis. And so if a self-replicating RNA molecule is ever discovered and there is a plausible naturalistic pathway for its formation, will that prove abiogenesis?
No, because RNA, even if we have an RNA wood, right? Even if we can make one in the laboratory, you will see that is useless for making the life we know of. Because as I mentioned, you know, you do not only need a genetic coding system, you need a decoding system, right? And those systems need to match. So a self-replicating RNA, for example, if we can make a self-replicating RNA, that does not mean that RNA will code a protein that can, you know, then can self-replicating or replicating the RNA at all. We have, you know, all genes will be transcribed into RNA and then translated into protein in all the lives we know of. And we know the RNA, you know, the RNA transcribed from the genes will have a totally unrelated function with the protein they encoded.
Right.
So there's no functional connection. The only reason RNA and the protein are connected now in current life we know of is because of our translation system. Our translation system will use the RNA as a template and determine, you know, where to start translation and what, you know, how to read the codes and how to, where to terminate the translation. So there's no functional connection. There are sequence connection because of our decoding system, but there's no functional connection.
Right, right. I think in the book you referred to RNA world, even if they could show there was a self-replicating RNA as a dead end, that it can't really go beyond what it is to our current DNA, RNA, protein system, as you say.
Right. So we have to start from the scratch again.
Right, start over, and so it's just a dead end. It doesn't, even if they could show it, it could possibly happen that you have a self-replicating molecule like an RNA, you still can't get to our biochemistry. Now, Rob touched on this, and that's the idea of needing these proofreading machines in the cell for DNA. So maybe could you talk a little bit about so let's say we could get to a self-replicating RNA or some primitive DNA system. What's likely to happen to that in the absence of the proofreading system?
The system breaks down immediately.
Right.
Right. It's very funny because the reason I laugh is, think about DNA replication, right? DNA replication, because we say, oh, that's why Watson and Crick was so excited when they figured out the double helical structure of DNA, because they said, well, immediately we see a mechanism of copying, right? But in reality, DNA cannot copy themselves. And actually, they cannot serve as a template even, right? DNA, as Rob said earlier, it's very long. In each of our cells, the DNA is like 6 feet long. And, they are stuck with each other because it's a double helix. They are stuck with each other. And you need to separate actually the DNA for them to be able to use as a template. And what nucleotides will be put, using the template, actually is not determined by DNA. The base parent, yes, the base parent is important, but the base parent is determined by the molecular machinery to do the copying. And the machinery, can, depending on different DNA polymerases, right? The copying machinery, where some of them will make more mistakes, some of them will make less mistakes. And so a big part to make the DNA replication, accurate is proofreading. So basically, you have kind of like if you are writing something, you have write with you a team, a team of workers checking to see your errors. And they will immediately, detect the error and help you to correct the errors, right? Which is very, very cool. Without that, without, the error correcting, you will introduce a lot of mistakes. And the more mistakes you introduce, the more mistakes will be introduced the next step. And the cell will just, quickly break down.
Okay, so in the miracle that even a self-replicating molecule could ever emerge, its future is dim, because it's not going to be able to endure all the errors that are going to occur. All the mutations will soon destroy that information that's necessary for replication, and it'll just fall apart again into its component parts, and you have to wait another billion years for another molecule to emerge that can self-replicate.
Right, DNA repairing system. Actually, there are more DNA polymerases involved in DNA repairing than the number of DNA polymerases involved in duplicating the genome. It's amazing, yeah. And like Rob said earlier, our genomes are dynamic, and a lot of DNA damages occurs every day in every cell of our body.
Okay, so I have some questions now. Someone says, "So very glad for your presentation. My jaw is on the ground. Thank you."
That's good to know. Very encouraging for me.
Another person says, "How is it possible to identify essential genes without knowing what they do?"
That's a good question, very good question. So by definition, essential genes are essential for the viability of the cells or its reproduction, right? So basically, you know, we can interfere the function of a gene by introducing mutations or introducing something to, you know, interfere the function without knowing its function, just to see whether when we do that, when we interrupt the function, even though we don't know the function, we will, you know, check to see whether the organism can survive or not. You know, for tiny microorganism like a bacteria or yeast, basically you just see whether, you know, there is a colony is forming. But since every colony will have many cells, so for those microorganisms, actually, you know, whether we're looking at viability or reproduction is hard to tell. Whether, you know, whether it's because they cannot live or they cannot reproduce, right? For higher, bigger organism like mice, we can tell, you know, whether we kill the mice, you know, or whether we interfere their reproduction. So it's a looking at the end result, right? We don't know the molecular details about what is molecular function are, you know, what process they control. We know the end result is that they're dead or they cannot reproduce.
Okay, thank you. John Harris asks, which steps on your model of the stairway of life are the easiest, relatively speaking, to traverse?
The first step, making the basic building blocks, right? So like synthesizing amino acids. That's what oh my gosh, I forget the name. What is that? Miller, right? Miller's experiment did, all right? So he basically, he just, you know, charged the methane, ammonia, hydrogen, and the water, and he got amino acid. Well, not all of the amino acids. And they cannot get, you know, pure chiral amino acid. So the amino acids he generated would be a mixture of left-handed and right-handed amino acids. And furthermore, the vast majority of his reaction products are not amino acids at all, right? So only a tiny fraction of his experiment generated amino acid. The vast majority are not amino acid, and you know, it's going to actually interfere the formation of life instead of facilitate of life. But we can find chemical conditions in the lab, you know, as organic chemistry. Oh my gosh, you're organic chemist. I did not know that, right? We know organically we can find synthetic pathways to make all the basic building blocks of life. Amino acids, you know, the nucleic bases, we can even synthesize a nucleotide, right? So that is the easiest step to crack. But what is interesting is that, you know, every step we move forward, right? Instead of finding that we're now getting closer to make a self-replicating cell, we are facing with a steeper step, right? you know, so instead of we're getting more hopeful, actually we're getting more depressed because the goal seems, instead of moving closer and closer, the goal is moving further and further. The more we know, the more we know where the goal is, so far away from us. It's just, it's impossible. Naturally.
Right. Yeah, I think there's a book entitled, I Don't Have Enough Faith to Be an Atheist, because these sorts of things like this. Well, I don't have any more questions now. So thank you very much, Change, for your talk. We really appreciate it.
It's a pleasure.
We're going to take a brief break until 11:15. So grab a cup of coffee or whatever, and we'll meet back here at 11:15 to hear from Brian Thomas. All right. Thank you.
I like it. It's very well organized. Thank you so much.
Thank you, Tan. Change. Okay, let's see. Now I need to go here.
Should I stop video?
No, I'm gonna make you an attendee again.